Introduce our keynote speaker, Sarah Gaffen. Sarah is one of the speakers I most look forward to hearing. And I first heard her speak what must have been many years ago at a fungal immunology meeting that was full of immunologists. And on a good day, I might be a mycologist, but I'm not an immunologist. And though I learned a ton of stuff at that meeting, I have to say it was a little bit like swimming in a foreign language until Sarah's talk, which was just so beautiful and so clear. I mean, you'll see today she has a true gift for clarity and a sparkling sense of humor. And she's a prolific scholar and a profoundly committed scientist, and I know we're in for a real treat to hear her talk. So after getting a bachelor's degree at Carnegie Mellon, Sarah received her PhD at Berkeley, working with Marion Koshland, and then she came to the Gladstone Institute at UCSF, working with Warner Green and Mark Goldsmith. She's been a full professor since 2012 at the University of Pittsburgh in the Department of Medicine, the Division of Rheumatology and Clinical Immunology, where she holds the Gerald P. Rodnan Chair in Medicine, and she's the Director of Basic Rheumatology Research. So as I'm sure you all know, Sarah's work is very well known. Her seminal discoveries on immunity to mucosal infections set a really high bar. From her work that first demonstrated that IL-17 receptor mediates oral microbial host defense, she's really deeply investigated the IL-17 signaling pathway and its role in mucosal immunity to fungal infections, as well as autoimmune disease pathogenesis and a number of other things. And also, I'm just going to tell you, you should look up her commentary in immunity on PubMed entitled Gut Busters, IL-17 Ain't Afraid of No IL-23. So I'm not going to tell you why you should look it up, but you should look it up on PubMed. You won't be disappointed. So Sarah's won numerous awards, including International Cytokine and Interferon Society BioLegend William E. Paul Award for Excellence in Cytokine Research, the Margaret Ragney Award for Advancements of Women in Medicine and Science, and most recently, she was awarded the 2025 Carnegie Mellon University Alumni Achievement Award. So Sarah, thank you so much for accepting our invitation to give the keynote talk. We're so happy to have you here.
Thank you. Thank you so much. I'm a redhead, so I'm really blushing right now. It's really an honor. Thank you so much. I've never been to this meeting before, and I now see why everybody is loyal and comes back again and again. I don't think it will be my last. One slight correction to that: if you do look up that paper, not the immunity version, look at the PMC version. They're different. Okay, so let's see if I can operate things here. I'm going to talk a lot about my favorite number, which is 17. And in fact, my 17th anniversary at Pitt is coming up in a month. So this is the amazing lab, one of those moments where everybody was in the room at the same time, and we took the opportunity to grab a picture. So when I say we, I really mean them. This is just actually a real street corner. It's not GPT. And you know it's Pittsburgh because there's the Steelers logo, and we all bleed black and gold. Okay, let's see if I can do this. And as I really like to point out, I'm a first-generation American. Everyone in my lab, and I dare say pretty much everybody in this room who lives in the States is either an immigrant or a descendant thereof. And I will not do further comment, but I think you guys know what I'm saying. Okay. All right, now I have to live up to my sparkling wit, apparently. Like everybody, we work hard. We try to publish our work in lots of our favorite journals, some of which are depicted here. But we in the IL-17 field are really lucky because we actually have a very special journal just for us. I have yet to get the cover of this, but I'm counting on this to be the moment. Okay. So I'm really a cytokine wonk. I still think of myself as a newcomer to the fungal immunology field, or the fungal field at all, which I guess I can't really say anymore. But I come at everything from the point of view of cytokines in immunology. And I always like to start with this. especially if you're not an immunologist, it's really overwhelming. All interleukin this and interleukin that. But if you think about them from the receptors, they kind of fall into a somewhat more manageable set of buckets. And I started working on IL-17 way back when I started my lab in the late 90s. Because it was clearly the founding member of a new family of cytokines that were structurally distinct from any others. And I thought, that's going to be really interesting to follow. There'll be, new discoveries to be made, and we can get traction in a place where there weren't a lot of people. And of course, in 2005, when Th17 cells got described, the world blew up. But under the umbrella of the IL-17 family are a number of ligands and receptors. I'm going to focus pretty much exclusively on IL-17, which is also IL-17A. I'm happy to answer questions about its close cousin, IL-17F. And these bind to a receptor composed of IL-17RA, which is absolutely essential for signaling through this receptor, and partners with another essential receptor, IL-17RC. But I want to point out, 17RA is also used by other family members, and there's other configurations of the receptor, none of which I'm going to bore you with today, but I'm happy to answer questions, and it's just something to keep in mind. So the Gaffen lab worldview is, of course, very IL-17 centric. And I'll talk a little bit about cells that make IL-17. This is certainly a huge area of field. Some people call it type 3 cells. I refer to them as type 17 cells. And these are all lymphocytes, but they include the conventional Th17 cells, which are CD4 positive, but also in some circumstances CD8 cells, gamma delta cells, the innate lymphoid cells, et cetera. After being made, IL-17 binds to its receptor, which I already mentioned, and activates various signaling pathways. And a big focus of my lab, really the through line of my lab, is the signaling that happens downstream of the receptor. I'm not going to go into that in great detail, but I think you'll see, again, knowing that, you'll see why we approach some of the questions the way we do. About half the lab focuses on autoimmune disease and the signaling pathways that drive it. I just will mention antibodies against IL-17 came into approval roughly 10 years ago and were really transformative for certain conditions, but completely failed in others. And so this has given us a lot of approaches in humans to understand what they do. I'm not going to talk about that really at all. Instead, I'll talk about what IL-17 evolved to do, which of course is to protect us. This will be all about candidiasis. Okay, probably don't have to really even show this slide. I think by now everybody is on board that Candida albicans is an important pathogen. As we've heard, it's a WHO priority pathogen and is a commensal in humans. I do want to mention it's not a commensal in mice, and mice are immunologically naive to this. I'll reiterate that a little bit later. And what kind of fascinated me when I first began to be exposed to this was the same organism causes all of these different diseases in very specific tissues, but the immune response seems to be quite different. And so systemic disease, of course, is why this is a priority pathogen, but oral candidiasis, really pretty much everyone with HIV/AIDS gets this, but yet they really don't get vaginal candidiasis, for example. So there's a lot of tissue-selective immunity, and this is a terrific organism to be able to interrogate that. So my talk will be divided into 3 parts, and hopefully I won't go too far over time. The first part, apologies to the Candida people who've probably heard some of this before, but a bit of the history of kind of how we got there and what IL-17 does, a little bit of the highlights, focusing on the work that sets up for some of the later and more newer studies. The second part will be, the second and third are brand new stories. I haven't spoken about them before. Looking at Candida, actually not a 17 story, it's a Candida signaling story in the oral epithelium, and then I'll end on our recent studies on vaginal candidiasis. Okay, so I want to show this just to get us all on the same page. This is a model that we've been using for a long time, and you'll see throughout the talk. Taught to us by Scott Filler, who is just a marvelous collaborator and incredibly helpful in getting us started. And really, I think, was my first encounter with just how amazingly, wonderfully kind and helpful the fungal community is. So this model, mice are infected sublingually with a cotton ball that's been soaked in Candida. And in order to interrogate fungal mutants, this is developed, mice are given cortisone because mice, like humans, have are able to fight this off and really don't have overt disease. So immunocompetent mice do just fine. So if you immunosuppress them, then they get disease, and then they clear it very rapidly. Now the only difference that we really did in this is we don't use cortisone because we're using knockouts, and so we're trying to more selectively interrogate immune components. Now, I mentioned before, candida is not a commensal in mice, and they are immunologically naive to this. And we and others have done a lot exhaustively to assess that. So these early immune responses that we see, mice usually clear by day 3 or 4, are all representative of the innate immune response. And I'll talk about that a little bit more in a minute. Okay, so for many reasons that I'm not going to go into, but in part inspired by the fact that Th17 cells were just starting to come on the radar, and that HIV patients are exquisitely susceptible to oral thrush, and the fact I was working at the time in a dental school, I thought it would be interesting to sort of look at IL-17 in various oral diseases, and one of them was oral candidiasis. And Heather Conti was a really spectacular graduate student who was working on something else, and she didn't like that project. And I said, well, you know, I had this crazy idea. You could look at oral candidiasis, but I don't know how to do it, and nobody in the building or the campus knows how to do it, and so you'll have to figure out how to do this in mice and blah, 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 thinking I would talk her out of it, which I completely failed to do. And what Heather showed was that mice lacking the IL-17 receptor were extremely susceptible. And Scott, as I said, Scott Filler really helped us get this model going, unlike wild-type mice that are very resistant. And you'll see data in a minute supporting that. So IL-17 receptor is important. So is its partner receptor, IL-17RC. We published that later. There's a key signaling molecule that's common to the IL-17 family called ACT1. It's also essential. And in mice that are very susceptible develop oral thrush that looks very similar to humans, both at a gross level and at histological levels. And I thought, great, we have a model system, we can interrogate IL-17, and we can go to town. But what was really gratifying was just a couple years later, Jean-Laurent Casanova and Anne Puel, who had cohorts of patients with chronic unexplained candidiasis, showed that a number of those patients had mutations in the same genes. So null mutations in IL-17RA, IL-17RC, later they found ACT1 patients, and various other genes that all converge on the Th17 pathway. So that told us not only did we have a nice system that happened to work in mice, but it was really reflecting what we see in humans. Okay, so over the years, we've used the system to really interrogate kind of, the who, what, when, where, why, how of 17 in candidiasis. It's an old review, but it actually from 2015, but it really still holds up. And so this is, now I'm showing you data. So wild-type mice clear candida. This is looking at CFU on day 5. And IL-17 receptor knockout mice have high fungal loads. And so that's the system, and it works. And so we've looked over the years at sources, target cells, and functions. And I'll be talking about these, but not necessarily in that order. So starting at the end, one of the things we found very early on from standard, actually back in the day when we did microarrays, anybody remember those? We found a bunch of genes, all of which have borne out, whether we do single cell spatial transcriptomics or RNA-seq, no matter what you do, however you do your gene analysis, we get a lot of the same genes, which include a lot of antimicrobial peptides, including defensins. And really the top hit was beta-defensin 3. That's the mouse equivalent of human beta-defensin 2, in case we have aficionados in the audience. And mice lacking this single beta-defensin are exquisitely susceptible to candida, almost as much as a 17 receptor. There's, of course, a lot of other things that get turned on, including some other antimicrobial peptides, also the neutrophil response indirectly through inducing chemokines. But I put beta-defensin 3 here because you're going to see that again and again throughout. All right, so that's some of what IL-17 does. What about where it acts? And one of the reasons we were so interested in this question is that as a signaling person, in order to study signaling, we want to study the right cells. So we really needed to know what were the target cells. And the IL-17 receptor, at least 17 receptor A, is expressed pretty ubiquitously. We had a lot of reasons to think it was an epithelium, and we had done a lot of things to rule out other compartments. But the oral epithelium is quite different from the skin, and particularly from what are technically called simple epithelia, like the gut. And anybody who works in the gut knows that simple is not the right term to use for that, or the lung, and these are all very distinct. So the oral epithelia is a stratified epithelium composed of a basal proliferating layer that has sort of stem-like properties, and this is characterized by keratin 14, which is in really all epithelial cells. And then this differentiates to create more intermediate and superficial layers that are characterized by a much more restricted keratin called keratin 13. And this is expressed in oral, esophageal, vaginal epithelia, and that sort of coincidentally or not are the tissues that Candida likes to infect. And so we created a Cree that would selectively delete in this superficial compartment, in part because this is also where we saw the dominance of IL-17 receptor expression. And this is very short. The Cree works. It's expressed in this superficial layer of the tongue. The pink cells here, that's this basal epithelium. So it's really very tight. These are available at Jackson if you want them. And so we use those, cross them to a flocked IL-17 receptor mouse, and you can see wild-type mice in black, they clear Candida by day 3. So I told you that verbally, but now you can see it. IL-17 receptor knockouts are very susceptible, and they stay susceptible forever, as long as we let them live. I think we've taken them out 5 or 6 weeks at least. And the keratin 13 deleted mice, so lacking 17 receptor, just in these superficial epithelia, were also very susceptible. And it's a bit, variable here. They're not quite as susceptible as a total knockout. But by and large, a lot of the signaling response, and especially the defensin response, really came from this keratin 13 layer. And that surprised us, because it's post-mitotic. You'd think these cells were really not very active, but yet they are still making really key antimicrobial proteins. More recently, Heather's lab has shown, interestingly, that platelets and megakaryocytes play an important role in Candida clearance, and the IL-17 receptor acts in these cells. This was surprising because us, as well as others, have done bone marrow chimeras, but as a warning, megakaryocytes are very resistant to radiation. So if you use bone marrow chimeras to tell you it's a hematopoietic cell, you're not actually hitting all of your hematopoietic cells. And I think this is going to be a really interesting aspect for her lab to pursue in understanding signaling in a hematopoietic system. Okay. So parenthetically, little aside, because everybody gets tired when it's Keynote, it's 4 o'clock in the afternoon, and I'm standing between you guys and dinner. When I was a graduate student, I worked at UCSF on IL-2 and T cells. And when I graduated, my mother, who's not a scientist, said, all those years, they read out my dissertation title, and she said, all those years you said you worked on IL-2, I thought you meant the bay where your lab bench was. True story. Okay, you can wake up now. All right, so where does IL-17 come from? And this is a little bit of an older story, but I'd like to tell it because I think it is the principles are bearing out in a lot of other systems. And so we did what every self-respecting immunologist does, is we ground up the tissue and looked at the T cells. The tongue doesn't have a lot of T cells, and it's a very harsh environment, so it took us a while to be able to get cells out, and there really are never very many. But anyway, when we did this, we found what we expected to see was that most of the cells that made IL-17 would be gamma delta T cells. These are lymphocytes at mucosal surfaces. They're sentinel cells. They had recently been shown to make IL-17 at the time, and they, that seemed like the most logical thing. And certainly they were there. However, more than half of the cells were expressing the alpha-beta T cell receptor and CD4. They looked for all the world like a conventional T cell. But I've already told you, mice are naive to Candida, or at least they were reported to be. And so we actually really probed into this in a lot of depth and showed in our colony, in our hands, as well as everybody else's, there's no pre-existing immunity in mice to Candida, no cross-reactive immunity to yeast and food, other Candida commensals, staph, anything, and that these T cells that we were seeing were highly diverse and they were not antigen specific. And in fact, if we used a TCR transgenic that had a a T cell repertoire to a single T cell, they could also fight Candida. So it didn't matter what the specificity of the T cell receptor was, and nor was there a requirement for ongoing TCR signaling. And we couldn't ignore them. I mean, I wanted to ignore them, but we couldn't because when you infect cells, it's the alpha-beta T cells that expanded. it's not that the gamma delta cells weren't doing anything, but they were not dividing, and certainly not in this early time frame. But these guys were, and you can see that here with Ki67 staining. So, I mean, there's also obviously a lot of questions you would ask. One of the first questions we asked was, what is activating these cells? What's making them what's triggering them to sense candida or to sense infection and divide? And so we took a yeast lock strain, and I was very proud of myself because I'm not a microbiologist. I was like, oh, I'm using a mutant. This is great. This is a yeast lock mutant. We've heard about it here. And we infect, and mice fail to induce IL-17, but a revertant does. And we thought, great. This tells us what we need to know, because this was not too long after the Gordon Brown and others had shown that the C-type lectin receptors sense beta-glucans, which are exposed on yeast hyphae, but not on Candida hyphae, but not yeast. So we figure, it's going to be dectin-1, or maybe some of the other dectins, and they all signal through card9. And there was some evidence for TLR, so TLR2 in particular. So great. So we took the time and money to, get the mice. It was my fiduciary overseer was not happy with that. But we got all these mice, and we infected them with Candida, and they all cleared. And the N is quite high here because we did a lot of mice. 17 receptor in mice do not. These guys are fine. And we went, okay, what the hell? So I don't know about you guys, but if I don't know what to do, I do a few things. One thing I have to admit I don't really do is sit down and read the literature nonstop because, denial. I was in Buffalo at the time, so, you console yourself with Tim Hortons. Any Canadians in the audience? Oh, yeah, there you go. My postdoc says he counts to 17 in all the languages that he knows, which is like 8. This is 17 in Chinese. Or you do what I did, which is to abandon your lab and move to London. So that's what I did for a year. And I did a sabbatical at King's College London, which is right behind the Shard, if you're familiar with London, and this is the Tower Bridge. And I worked with Julian Naglick. So Julian, I was there at a really opportune time. It was when Julian and Bernie Hubert were in the process of finalizing what turned into one of the most seminal papers in the Candida field, which is the discovery of Candida lysine. And they were trying to understand the response of oral epithelial cells to Candida when it becomes hyphal, namely dangerous. So Candida lysine is only in hyphae, not required for filamentation. That's important. So they form filament. Even if they don't have it, and it's a secreted pore-forming toxin that is secreted in the invasion pocket and triggers local damage in the epithelium, which then responds by inducing a program of innate immune cytokines, including IL-6, IL-1, CCL20, beta defensins, and this is a very similar program to what IL-17 will induce in epithelial cells. These are epithelial cells; they do not make IL-17, but they're acting in a sort of a parallel manner. And so we thought, well, wouldn't it be interesting if this had something to do with activation of the innate response? And sure enough, if we infected mice with an EC1 deficient Candida, IL-17 expression was basically not turned on. Same thing for IL-22, another TH17 cytokine, but the revertant induces it just fine. And this is dependent on production of IL-1 and IL-36 from the epithelium. And I think what this is telling us is that, and this is sort of relating to the last talk we just heard this morning, this is a way for the epithelium not just to see yeast versus hyphae, which is what dectins would do, but really to distinguish between hyphae that are damaging and those that are not. And various strains of Candida express different levels of Candida lysine. It's really related to how virulent they are. And so it's really sort of a more subtle and nuanced way to sense danger, I think. And I think this is a general principle. These papers came out not too long after ours, showing that in staph, for example, which secretes a pore-forming toxin, triggers similarly production of IL-1 and IL-36, which amplify innate production of IL-17, in this case from gamma delta cells dominantly and innate lymphoid cells. Also Candida lysine can trigger platelets through pore-forming reactions to trigger a type 17 axis in the lung, is work by David Corey. Okay, so type 17 cells don't just make IL-17, they make other cytokines like IL-22, and these are siblings, not twins. They activate quite different signaling programs. IL-22 is a classic JAK-STAT activating cytokine, and IL-17 is really much more like IL-1 or TLRs and turns on NF-kappa B and sort of acute signaling. And so Felix Agora in my lab decided to pursue IL-22, and you can see IL-22 knockout mice or IL-22 receptor knockout mice are very susceptible to candida. They fail to clear it, unlike wild-type mice. Together, 17 and 22 have much more cooperative effects. This is at least a log increase, and those mice actually end up dying, but not by disseminated candidiasis, interestingly. And of course, we duly asked, where does IL-22 act? Being absolutely sure it was going to look just like IL-17. Reminder, IL-17 acts in the superficial layer, K13 positive. Absolutely no contribution of IL-22 in the K13 layer at all. I think there's at least 30 mice in this experiment because Felix just kept doing it. And by God, they are not susceptible. But then, Felix is a really smart guy, and he came to me and he said, yeah, maybe there's a little 22 up here, but it's really in these basal cells. And so when we crossed it to a keratin 14 deficient mouse, and this is a tamoxifen inducible version, the mice were susceptible. The knockout wasn't complete, so it wasn't quite fully susceptible, but it was certainly there. And we saw exactly the same thing for STAT3. And we'd actually been sitting on the STAT3 keratin 13 deletion for years and couldn't figure out what it was doing. But STAT3 is also needed in this compartment. And so what that said was that even though 17 and 22 are made by the same cells, they're really acting on different layers of this stratified tissue. And whereas IL-22 is driving a proliferative event on this more basal cells, triggering them to differentiate and to essentially replenish this more superficial layer. And by doing that, it essentially licenses this tissue to respond. And I've got a question mark here. So 22 is acting here and 17 is here. What's downstream of 17? Hold that thought. Okay, so 22 through STAT3 drives this proliferative program, replenishes and heals the tissue. This is all put in place by candida lysin, which mobilizes activation of these cytokines through type 17 cells. Okay, so I raised the question, how is IL-17 doing what it's doing? This is what we always think about. And the sharp-eyed among you will have noticed that I had totally blipped over this bright yellow transcription factor, I-kappa B zeta, which is induced not only by IL-17, but also by IL-22. Now, I-kappa B zeta, don't be fooled by the name. It's not an inhibitor. It's an activator of transcription. It's actually a co-activator, so it interacts with other transcription factors to drive transcriptional responses and also epigenetic responses and remodeling. It's induced very potently by IL-17, but we've also shown it's induced by IL-22 in epithelial cells, but also IL-1 family members, TLRs. And it's only induced, interestingly, by virulent candida. So if this is, these are oral epithelial cells in vitro. This is SC5314, apologies, this is the most virulent strain of candida that we've been using. Induces I-kappa B zeta, encoded by the NF-kBIZ gene. And this fails to be induced by either by a candida lysin deficient or by other avirulent strains of candida. The total knockout of I-kappa B zeta was problematic because the mice had a lot of systemic issues. They were very susceptible to staph, so they would pick it up in the facility, and they, we think, even have maybe some real developmental issues. So we couldn't really get at this until we got our hands on floxed mice, which we crossed to a tamoxifen whole body inducible, so a ROSA 26. This allowed us to delete the gene entirely, but at will. So we did this a week or two before infecting mice with candida, so we bypassed all those early developmental issues. And the mice were very susceptible to candidiasis when we did that. And the tamoxifen controls are not. If anything, they're even more susceptible than a 17 receptor deficient mouse. And so, of course, we immediately asked the question of, is it acting in all the same places, or does I-kappa B zeta have a more broad effects? It certainly acts in the keratin 13 layer again, so very susceptible. No effect in the keratin 14, which surprised us because we thought that was going to account for its increased susceptibility and the fact that it was induced by 22. But it has no discernible effect in this basal layer whatsoever. and certainly we think is at least part of the explanation for what's down of IL-17. This is not a 17 signaling talk, but we've ruled out a lot of other transcription factors that are really important in autoimmune disease driven by 17 that have no bearing in the candida response, which is sort of interesting. So this is really the main one that we found so far. Okay, and one of the things it does is to serve as a coactivator for gene expression of the defensin. So it binds to the beta defensin promoter and allows its activation. And I just wanted to give a shout out to this really nice new MBio paper by Priya Uppaluri, who's at the Lundquist Institute, who did this really nice spatial transcriptomes atlas of OPC. So anybody who does OPC, I encourage you to look at this. These are uninfected and infected tongues. This is cut through the middle. I forget what term that is. And it's really cool because you can see the candida RNA as well. So this is candida in black, and it's around the periphery, and then you can really see all the tissues, but also the genes. And I'm just pointing out, here's my favorite, I kappa B zeta, right there. But there's a whole lot more in this paper, so I encourage you to check it out. Okay, so to summarize this, and a lot of stuff I haven't told you, I've shown you that when candida becomes dangerous, and dangerous is really defined by producing candidolysin and triggering damage, this triggers production of IL-1 and 36, I didn't actually show you that, mobilizes type 17 cells that make 22 and 17, which signal through their respective receptors on their respective target cells through their respective transcription factors, leading to a program of anti-candida immunity that includes chemokines. I really didn't talk about this at all, contributing to recruitment of neutrophils, but also to regulation of defensins. And this cup that you can buy on Cafe Press, I had nothing to do with this, I swear. Somebody came across it and they said, "Oh, Sarah, was this your design?" And it's like, I am not that artistic. But I did buy the t-shirt. Okay, this next story is a brand new story. We just put it on bioRxiv, I think on Sunday. And this is work done by Nicole Pond, who did her PhD in Julian's lab. I met her when I was on sabbatical there, and she's now postdoc in my lab. And this is really about signaling responses in epithelial cells to candida. So we can forget 17 for the moment. And what Julian's lab has done and Nicole has contributed to is really looking at all these various transcription pathways that are mobilized when you have candida in its high full state. So ignore all the details. But there's really been a lot of really beautiful and elegant work done by Julian, by David Moyes, who is now independent, looking at how all these pathways are activated and the transcription factors that regulate target genes being in particular cytokines and chemokines. So we know a fair amount about the transcriptional signaling pathways that are triggered. This is epithelial cells, but of course people working in macrophages and neutrophils have really looked at those as well. So there's a lot done about the transcriptional program. But what's sort of surprising to me, in my other half of my lab, we do a lot of work on post-transcriptional signaling. And as an immunologist, immunology, target RNA, these are so unstable. It's a pain in the ass to work in immune genes because if you look at them cross-eyed, they degrade. And so there are a lot of mechanisms that are in place in the immune system, the body in generally, to control RNA post-transcriptionally, particularly at the level of stability, but also translation. A lot of checkpoints there. And yet very, very little had been done in this space of looking at this. So Nicole decided to take this on. And one of the things that we decided to look at, or she kind of was excited about, was something called epitranscriptomic regulation of RNA, or modified. And now I have to say, I sort of felt like a total dingbat in the sense that until about 5 years ago, I didn't actually know that RNA was methylated. And I was like, I was telling anybody who would listen, did you know that RNA is methylated? And they're like, yes, we did, Sarah. Where were you in biochemistry? And the answer is the bar up the hill. Anyway, so RNAs, and of course, with the pandemic and the verboten RNA vaccines, we know that methylation or modification of RNAs is really important to its fate. The most common modification is N6-methyladenosine, and as much as half or more of RNA transcripts have at least one methylation site. And it's especially enriched in 3' UTRs and helps dictate stability, both stabilizing and destabilizing effects. But this process also regulates RNA fate in a lot of ways. It's quite complicated, but there are writer enzymes in the nucleus that deposit these, and this is contemporaneous with transcription, so you get methylation of RNAs. And this is dynamic, so it's not it can be reversed, and there are demethylases, or so-called erasers. I won't talk about those at all. But really, the main part, at least of this talk, will be the readers. So these are the RNA binding proteins that recognize either methylation or the changes in RNA structure that occur as a result of methylation. And there's a bunch of them. And these really, particularly those in the cytoplasm, will dictate the consequence of RNA. And we had been looking at this completely in a different reason in the IL-17 pathway, and so we were starting to know how to kind of approach this. But of course, it was like, well, has anybody looked? Surely somebody's looked at this before us. So this is a pretty recent search. RNA methylation in Candida, we got, did you mean DNA methylation in Candida? No, I didn't. And then if we did M6A in Candida, there's two, but they're the same paper. And, I don't know, I'm not familiar with this journal. It's not that nobody's done anything on this pathway in infections. So there was a handful of papers looking at M6A in fungal keratitis. But really, as you can see, nothing with Candida. There has been quite a bit done in the viral space, so looking at interferon regulation and regulation of viral genes, and some studies in bacterial and parasitic infections, but really, surprisingly, not a whole lot. And so Nicole decided to undertake just a sort of relatively simple siRNA screen to see, is there a there? So she took oral epithelial cells, the TR146 cell line, it's a human cell line, infected them with Candida yeast, and for 4 hours later, she played with time points, but this was a fruitful one. Candida has become hyphal, and she just looked at a few of our favorites, and so just some of the representative genes. And then knock down different classes of readers. So just to save you a little of a headache, we ruled out the class of imps, which is the ones we were kind of excited about because we had all the mice and we had been studying them for other things, but we didn't see anything. The YTHDCs. But she did see some hints of effects when she knocked down a class called the YTHDFs. And so this is a triple knockdown in TR146, just looking at a couple of representative genes. And all of these genes seem to be somewhat up, suggesting that the YTHDFs had a destabilizing effect on these RNAs when it was present. And that's not surprising because that is the main function of YTHDFs, or a major function of them. So she then looked in RNA-seq to look a little bit more broadly. This is not the entire list, but this is a subset. And what was interesting was that some genes were definitely up, like some of the ones I showed you, but a lot of genes were also down, including some of the antimicrobial peptides we've become interested in, the so-called small proline-rich proteins, or SPRRs. And here we've got chemokines. EGR2 is a transcription factor that's been implicated in Candida infections. And so there certainly seemed to be a lot to play with here. But those are just genes that are changed. Are they really methylated, and what happens upon infection? And so looking at the SRAMP database, which really tells you about methylation, a lot of these, pretty much every gene she saw was had a predicted methylation site. And then she undertook what I thought was going to be a really challenging technical issue, which was to do RNA IP with antibodies against M6A. Turns out that the available technology, the kits, work really great. And so this is TR146 cells, and what we're doing, if you pull down IgG, you don't pull down CXCL8. Uninfected, we don't pull it down. There is quite a lot at baseline, but we don't pull any more down, but it's highly enriched after infection. And this is true not only in a cell line, but remarkably, even in vivo. She was able to see this in tongue, and this is just a few predicted ones. This is one of the SPRRs, again, highly enriched, and we're now looking at this a little bit more globally. Okay, so they're methylated. If you knock down some of the YTH, some of the readers, then the genes go up. Does this do anything in vivo? And because genes went up and down, we didn't know what to predict if we could block it. We could not simply use knockouts because there are not flox YTHDF mice. and the genes are they're on 3 different chromosomes, and the total knockouts are lethal. So, it's going to be a very long-term project to interrogate those. But fortunately, there is actually a first-in-class drug that targets the writer at metal-3. So this is the core writer complex, and this has been used to look at AML models. It's under very serious consideration as a drug target. And when we treated mice and subjected them to OPC, they did better. So if we look at fungal loads here, particularly in the early time points, fungal loads were substantially lower. But it was really obvious when we looked at clinical signs, including weight loss. Mice, these are wild-type mice, which are destined to clear candida in 3 to 5 days. They do lose weight in those early time points. But if you treat them with the inhibitor, they don't. This is not prophylactic treatment, this is pre-treatment, so we don't know what happens prophylactically, but it's really quite impressive. And as I said, associated with drops in fungal load at those time points, and increase in a number of the predicted targets like beta defensins, also SPRRs and many others. So to conclude this section, RNAs that are induced by candida are subject to methylation in oral epithelial cells, doubtless in other cell types as well, and that there is potential for targeting this pathway in a therapeutic way, and we're certainly trying to pursue this in a lot more depth. Okay, last part, and then we can drink. So I've told you a lot about oral candidiasis, and for historical reasons, that had really been a lot of our focus. But vaginal candidiasis is actually by far the most common manifestation of candidiasis, and it's an estimated 75% of women, and I frankly don't believe that. I think it's way higher. I have yet to meet a female that has not been afflicted with this, and it can be recurrent, as many as 4 or more times a year is defined. Really serious impact on quality of life, high medical burdens, and a very neglected and underdiagnosed disease. It's different from other manifestations of candidiasis in the sense that this is really defined not so much by how much fungus you have, but by the immunopathology associated with it that really causes symptomology. And it's tightly linked to estrogen, which I'm going to talk about, but also other factors, including pH, et cetera. Microbiota, particularly lactobacilli. Unlike other forms of candidiasis, it is not an obvious manifestation of immune deficiency. So HIV-positive women do not get vaginal candidiasis at high rates, unlike oral, which pretty much everybody with low CD4 counts encounters. Nor is it associated with Th17 impairment. However, there's good evidence that there's adaptive immune responses that work because in preclinical models, it can protect, and even the most advanced Candida vaccine was trialed in VVC. And there was definitely evidence of efficacy, particularly in recurrent VVC. And that was all halted, not for any scientific reasons, but really lack of money and political will. There is a very strong type 17 gene signature induced in VVC, so many people feel that tells us that it's involved. But IL-17 knockout mice, the ligand knockout mice, were shown by Paul Fidel to be resistant. So it's really kind of still controversial in the field, an open question of whether the 17 pathway has anything to do with it. And I wasn't really touching this at all, but Brian Peters came to me, he's really a leading expert in this, and said, I want to come to your lab, you've got all those mice, I want to run I'd love to run your mice through this model. And I said, great, you have to teach it to us. So he came and spent like a month in the lab and taught my lab manager, Bianca Coleman, who's really led a lot of this, the model. So very briefly, I said this is an estrogen-dependent system to condition, so wild-type mice are pretty resistant, but if you give them estrogen, then they become susceptible. So we give them estrogen, you collect vaginal lavage fluid, you can also collect vaginal tissue. Everything I'm going to show you is VLF, but we see the same thing in tissue at early time points or later, day 3. Okay, so let's start with estrogen, that's the gray bars. So if we take IL-17 receptor knockouts, which are exquisitely susceptible to oral candidiasis, there's no real difference, wild type or knockout. And the same was true with an IL-22 knockout, really the same. And the same was true, we threw our K13s through it, what the hell. ACT1 knockouts, 23 knockouts, 22 receptor knockouts. Nobody showed increased susceptibility over wild type. And we also did this without estrogen because, I don't know, nobody does that, but I don't know. I didn't really know what I was doing. I was like, well, if we always got to do your controls. So we did that, again, nothing. However, People always see this, and they're like, yeah, but look, it's not statistically different. It doesn't matter what statistical test you do. You do the right one or you do the wrong ones, doesn't matter. You can't make it. I tried, trust me. Okay, so we published the paper. Oh crap, I don't know how to back up. Here we go. And I'm going to live to regret this title because we're saying it's dispensable for VVC regardless of estrogen. Okay. However, a beautiful meta-analysis was done. I'm normally not a fan of those things, but this is a really reputable set of people. They studied 17,000 patients who were on anti-17 therapy. And not only was oral candidiasis a very notable side effect, some people say that's not true, but clinicians will tell you that, but it is, they do. But VVC was also a very modest, but absolutely statistically significant side effect. And I'm in a rheumatology department. My colleagues say, yeah, we see this. So there was that. We had that little hint in the 17 receptors. And Melissa Cook was looking at vaginal cell responses to cytokines, and she found that if we took, this is a human vulvar-vaginal cell line, so it isn't immortalized. We'd love to do this with the more primary cells. But if you treat them with IL-17 and 22 alone, you don't get a whole lot of response, a little bit. But if you put them together, really strong synergistic responses. And I'm showing you, this is the human analog to beta defensin 3. This is BD2. But we saw this for a lot of genes, suggesting maybe that there's cooperative effects. And because we already had these IL-17 receptor, IL-22 receptor double knockouts, we put them through the model. And of course, I wouldn't have given you all this buildup if we didn't have a result. And we were really pretty surprised. So again, I'll start with the estrogen-treated animals. The wild-type mice, very susceptible, as we always see. The double knockouts are even more so. And again, this is a log scale. And what that told us was a couple things. That the 17 first of all, there's more to go. Estrogen definitely renders mice susceptible, but there's room for other signals that are obviously protecting them at some level, and those signals are driven by 17 and 22. So it suggests, there was a hint that these are two different pathways. But really the big surprise was if we don't treat with estrogen, they're still really, really susceptible. I mean, at the level as they would be with estrogen. Again, to me, implying that these are separate pathways. And I will show you in a second that estrogen is normal in these mice, because that was sort of the obvious question. And this is not just fungal load, this is also immunopathology. You can see the histopathology here. The LDH scores are remarkably higher. Interestingly, PMNs don't go up. So there's no change in the number of neutrophils, even though that's sort of often used to define disease, but there's really absolutely no difference. And they're obviously not clearing candida. There's a lot of cytokines were elevated, including IL-1, which I'll show you in a minute. So this is not only fungal loads, but also immunopathology. Obvious question, well, maybe they just have high estrogen, and therefore you're rendering them more susceptible, but we're not, so estradiol is normal. Progesterone, interestingly, is consistently elevated. There's no change in all the other hormones that we looked at either. They are fertile, they have normal estrous cycle dynamics. I've just depicted the mouse cycle here, and as I said, that's normal. So because progesterone was higher, now progesterone's been tested by Paul Fidel, and if anything, that actually protects mice. It certainly doesn't render them susceptible. But what it does is it drives them out of estrus. So we treated mice with progesterone or estrogen, so just showing that we can do this. These are pap smears. So if we give them progesterone, aka P4, they're not in estrus, and they're still more susceptible. This is day 3 and this is day 7. And they still have increased fungal damage, and the PMNs don't change. So this, again, I think really supports the idea that what we've uncovered is an estrogen-independent pathway that is driven by both IL-17 and 22, and you need them both. Okay, we have a minute. All immunologists want to know what's upstream. I already mentioned that IL-23 doesn't. Activation of both innate and adaptive type 17 cells involves IL-23, but also IL-1, depending on the setting and the situation, et cetera. IL-1 receptor knockout mice are also more susceptible, both with estrogen and without. And if we look at the genes that are affected, it's really type 17 genes. Here's 17F, 17A, 22's on here somewhere, beta defensin 3, all of our favorite genes. This is work done by Brian Peters. And however, even though IL-1 is driving type 17 cells. IL-1 alone is not enough because the double knockout mice have really high elevated IL-1 and a perfectly intact IL-1 receptor. So IL-1 signaling is important, but it's upstream of 17, not downstream. Okay. Oh, wow. I think I did pretty well on time. So to summarize that, we think we've uncovered, we think at some level we've reconciled some of the debates in the VVC field. It's not 17, it's 17 and 22. And so this is distinct, but yet maybe not completely distinct from immunity in the oral cavity, where these cytokines really drive a protective response. We're working on figuring out all the details of what that protection really is. This is mobilized by IL-1, which is upstream, not downstream of this pathway, and we believe is independent of the estrogen response. And I've really learned a lot here from people who are doing a lot of studies in the estrogen-dependent signals, so this has really been great. I'll let you guys read this. I'm delighted to take questions with the 7 seconds I have left. I'm really proud of that. And I will encourage trainees to be brave. So thank you.
Questions?
And if there's any fungal people in the audience, I was given words and I got them all.
This is a really interesting work. I'm curious about whether or not you've looked. I know you in the last kind of like set story where you saw similar neutrophil numbers, despite differences in, I guess, pathogenesis. Have you looked at all at any like markers of activation, like on the neutrophils that may like account for some differences in inflammatory like responses too?
We haven't done that in great detail, but what we have done is deplete them, and there's no difference. And that's been done in VVC before. That PMNs, you know, they're essentially, people describe them as anergic. In part, that's due. Paul Fidel's lab has shown heparin sulfate can help suppress their activity, but we're absolutely probing that in more depth. You know, I think. But the data are kind of pointing away from them, so we have looked at sort of some of the kind of standard markers, and they really don't seem to change. So yeah.
Hi, great talk. So in case of the IL-17 and IL-22 double knockout, so do you see, is it a systemic IL-1 beta upregulation, or is it a particular cell type?
Do we see what?
So is it a particular cell type that contributes to the increased IL-1 beta, or it is a systemic like all the cells?
So who's making IL-1? We don't know. I mean, IL-1 can be made by pretty much every cell. If I had to guess, I would say that it's probably the epithelium, but it could absolutely be all the cells. So yeah, I hope the reviewers don't ask us to do that. Or maybe they will. I mean, it's easy to sort of at least like start to parse it out, but yeah.
The RNA methylation writer that you talked about, is its activity or its production regulated depending on the activation state of the cell? Yeah,
it's a great question. So we have no evidence yet that the writers or the erasers change levels. The activity is a little bit harder to assess, especially for genes that are, when the genes themselves are going up. You know, is it simply a product that there's more RNA and therefore you're seeing more methylation, or is the ratio different? So we are trying to probe that. We have hints that ratio is different, suggesting there might be increased activity, but it's quite indirect.
Any other questions for Sarah? Okay. Oh.
Hi, really nice talk. I was wondering what signaling is more important. Is it the IL-17/22 or is it estrogen? And can you block estrogen and use your knockout mice and you get rid of that susceptibility?
Yeah. Okay, so the technical challenge of blocking estrogen with tamoxifen is that tamoxifen is also anti-candidal. So one of the things, when we use these tamoxifen-induced knockouts, we actually, every mouse in the experiment is getting tamoxifen to make sure that isn't an issue. We tried using, oh God, I forget what it's called. it's one of the aromatase inhibitors that can inhibit production of estrogen, and the experiment turned out to be really messy and wouldn't really tell us anything. So what we're going to have to do, and we just got the protocol approved, is overectomies, right? So you do an overectomy, so then the mice can't produce estrogen at all, and really to nail that. So yeah, we're now allowed to do it. That was kind of why we went to the progesterone experiment, was to sort of essentially get at the same question in a kind of different way.
Thinking about the second story, I was wondering, so candida probably also has readers and writers and all of this similar enzymes. So does the inhibitor also have any effect on candida itself, or do they try to like send something back and forth to do it?
Yeah, no, absolutely.
Really cool.
Absolutely have to do that, but we haven't.
Everybody wants it now, so.
Hey there, Sarah, great talk.
Oh, there you go.
So in the human data that you've looked at, going along with the third story, is there any data that supports increased risk of vaginal candidiasis with estrogen suppositories? I'm channeling in that IL-17 antibody-treated population. Because I'm thinking, my wife's
not to my knowledge. It was a great question.
Yeah, my wife's a gynecologist and a huge estrogen suppository evangelist, so I'm wondering if maybe there's an opportunity to even try to model that within your mouse model to some degree.
That's a really interesting idea.
Maybe even within like geriatric mice to some degree that maybe reflects that.
I don't want to do geriatric mice.
No, I mean, of course not. I mean, you were talking about expensive mice.
Talking about fiduciary responsibility.
Yeah, exactly. But
no, that's an interesting idea. I mean, in some senses, you could say that the estrogen-treated mice is sort of mimicking that. But
right.
Yeah. But the human's interesting. To my knowledge, no. But